Welcome to the first of our three-part series on proteins. Proteins are so amazing and spectacular and fabulous and ubiquitous that it takes not one, not two, but three screencasts to cover the entire subject. So here we go. Proteins include a diversity of structures featuring in a very wide range of functions. Again, structure, function, structure, function. Okay. Proteins account for more than 50% of the dry mass of most cells. Again, most of it is water. But there are many, many, many different types of proteins with a variety of functions, and therefore, of course, a variety of different structures. Protein can be structural support, storage, transport, cellular communications, movement, and defense against foreign substances. So let's just take a quick peruse through some of those things. So we've got proteins that are enzymes, so they accelerate chemical reactions. We have proteins that are defensive, your antibodies, you guys have heard of those, help with your immune system. Proteins that store stuff, they store protein, they store energy, they store all kinds of good stuff, they store food. Proteins that are transport proteins, they move things around, they move things within cells, they move things in and out of cells. We have proteins that are hormones. Not all hormones are steroids, right? Testosterone, estrogen, those are steroid hormones. But we also have hormones that are proteins, like insulin and glycogen. There's insulin. Receptor proteins that help with communication and help your cells talk to each other. Contractile and motor proteins that help you move, because we like moving, not like those plants. Structural proteins that basically help hold you together. Collagen is a support protein, kind of helps your body from like flying apart into a million pieces. So let's learn, actually, before we go. So one special, and most people, the most common function people associate with proteins is an enzymatic function. And so enzymes are a very special type of protein. They're kind of like the rock stars of, the protein family, the movie stars. Everybody's like, oh, enzymes, and nobody cares about the poor little structural proteins. But they are amazing. So they act as catalysts to speed up chemical reactions. And we have a whole entire chapter on enzymes, so they will be coming soon. All right, so let's get back to take a couple steps back and talk about exactly what is a protein, how do we make it, what's it made of, etc. So proteins are also called polypeptides. And they're a polymer, a long chain of peptide bonds. And they are unbranched polymers built from the same set of 20 amino acids. So amino acid is the monomer of proteins, okay? And a protein is a biologically functional molecule that consists of one or more polypeptides. So what exactly does all that mean? So let's look first at the monomer. The monomer that all proteins are built of, all polymers are built of proteins, is called the amino acid. Okay? Amino acids are organic molecules, and they have a carboxyl group, which is where the acid comes from, and an amino group, which is where the amino comes from. As we said, there are 20 different amino acids, and they differ in their properties due to differing side chains or functional groups, and these are called R groups. So the next slide shows us the structure of the basic amino acid, and all amino acids have the same basic structure. So they all have this central carbon. They have an amino group on one end and a carboxyl group on the other end. This is the amino. And remember, carboxyl groups in water will ionize into carboxylic acid groups. So that's the acid part. Okay, so amino acid. Then, so this part of every amino acid is the same. Okay, then what makes our 20 amino acids different is this R group or side chain. And R is not an element. So this R is like a representative letter for whatever that side chain is. So let's look at some of the different R groups that we can have. And oh, whoop, there they all are. So here are all 20 amino acids. And you'll see that this part of every amino acid is the same, the amino group, the central carbon, and the carboxyl group. And then the R group is what makes them different. Okay? So we're going to actually zoom in on each of these ones. We'll start off with these, the nonpolar, then do the polar, and then do the charged. So here are my nonpolar side chains. That means the R group is nonpolar, and they're all the R groups are hydrophobic, okay, which makes the amino acid hydrophobic. So just straight up hydrogen, there's a methyl group, here's a whole bunch of fanciness. Oh my gosh, look at this ring, blah, blah, blah. So all of these R groups, if you look at them, they're mainly composed of hydrogen and carbon. So they are nonpolar R groups, and they make these amino acids nonpolar and therefore hydrophobic. We also have another set of amino acids that have polar side chains. And these guys are therefore made hydrophilic because the character of the amino acid is determined by the R group. Okay, so if you're trying to determine what type of amino acid you have, whether you have a polar, nonpolar, look at the R group. So here we've got some hydroxyl in that R group. Here's a sulfhydryl. We got some aminos in here and some more oxygen. So all of these guys are polar. So they make the amino acids hydrophilic. Then there's a couple of amino acids that are even fancier. These guys are charged. Since they're charged, they are, of course, polar and hydrophilic as well. So we have some that are acidically charged, and these guys basically have that carboxylic acid group on the end. Okay, so those are acidic. And the ones that are basic or positively charged have an amino group on the end. Amino group, amino groups, amino groups. Okay? All right, so again, the character of an amino acid or the flavor is determined by the R group because it's the only part that's different. They're the same in this section, right? So how do we go from amino acids to a protein? So amino acids, again, are the monomer. And to link them up into a polymer, we use a dehydration synthesis reaction. And the bond that is formed between amino acids via a dehydration synthesis reaction is called a peptide bond. And that's where we get the name polypeptide. So a polypeptide is a long polymer of amino acids linked by peptide bonds. And again, the different structure, the different function is determined a lot by the length of the polypeptide chain. So it can just be a few amino acids long to thousands and thousands of amino acids long. And this is what allows, because there are 20 different amino acids, this is what allows for basically almost infinite variety in the structure and therefore function of proteins. Every polypeptide has a unique linear sequence of amino acids. And on one end, there'll be that carboxyl group, and that's the C terminus. And on the other end, there'll be an amino group, and that end is called the N terminus. So let's have a little look here. This is how we make a polyp. So I have here 1, 2 amino acids here, and they are linked together by a peptide bond. To add more to the polymer, we have our hydroxyl group here from the carboxyl group, hydrogen here from the amino group. We subtract out our water, and they are replaced by a peptide bond, and you extend the polymer. And then you just continue to do the same thing, keep with the dehydration synthesis reaction to form the next one. So you would kind of just add it right here, plus another one, H, N, H, C, C, etc. You got it. et cetera, and so on. That really is messy. I'll draw it better in class. Okay? And that is the basics of proteins. They're made of amino acids formed by dehydration synthesis and synthesis of peptide bonds. So end part 1. On to part 2.